And now, an exclusive sneak preview of a recent X-Talks webinar, Enhancing Treatments Using Targeted Protein Degradation.
Traditional drug discovery primarily focus on inhibiting the biological function of target proteins, where blocking the functional or catalytic pockets. This strategy requires that the target protein to be accessible, and considerations must be made for affinity, specificity, and activity. There are clear limitations to this approach. For instance, only an estimated of 20% of cellular proteins are predicted to be potentially targetable. Furthermore, issues such as side effects and rapid resistant foundations are also challenges that need to be addressed. To overcome the limitation of old-fashioned designs, scientists are actively exploring new strategies for drug design, one of which is the most promising approaches involve leveraging the protein's degradation pathway within cells. In eukaryotic cells, proteins can be eliminated via two pathways, the proteasome and the lysosome. In proteasomal degradation, the protein substrate is recruited by E3 ubiquitin ligase, and then following with ubiquitination and subsequently digested by the proteasome. Lysosome, on the other hand, are responsible for the degradation of surface proteins and large particles. Correspondingly, drug discoveries that target proteins of interest and trigger their degradation, where these two pathways are evolving. Its concept is to use bifunctional molecules to link the target proteins and the degradation machinery together, thereby switch the degradation. Today, we'll focus more on a hotspot in drug development strategy, the ubiquitin-proteasomal degradation, in short UPD. Currently, there are three kinds of UPD molecules are developed following the UPD pathways. Molecular groups, PROTACs and ABTAC. The molecular group is a bifunctional molecules that itself can bind both to E3 ubiquitin ligase and the protein of interest to bring them together. And PROTAC is the hetero bifunctional molecules with two functional groups which binds to E3 ubiquitin ligase and the protein of interest respectively to bring them together. And the most emerging technique, the ABTAC, is using the bio body to bring the protein of interest, the transmembrane or cell surface protein of interest, together with the transmembrane E3 ubiquitin ligase to trigger the degradation.
To view the entirety of this webinar, visit the link in the description and register on xtalks.com, completely free of charge.